Good afternoon, everyone. And this is Dr. Samuel from the Department of Biotechnology. And hope this is the right time for us to share information on the drug discovery process or drug discovery pipeline. An essential topic to be known and to be understood by everyone, because it occupies a significant area with respect to your marks, right? Drug discovery, an essential topic in the unit 5 of the paper called Genomics and Proteomics. And it should be expected by you in the section C, right? It is a definite question. Explain the drug discovery pipeline, right? So be attentive and be online to capture the things I'm going to tell you. Fine, let's move on to the topic. What is drug discovery, or what is drug? See, drug is an entity prescribed by a doctor for a disease, right? Drug is an entity prescribed by a doctor for a disease. And the drug may be from biological origin, or it is being synthesized through chemical process. So irrespective of their origin, its role is to bind, interact and modulate with the receptors of cells, right? Just note down the points to be or the role to be carried out by a drug. A drug should interact, a drug should bind, and the drug should modulate the receptors or the proteins expressed on the cell surface. An essential role. Fine. So I told you a drug is synthesized either by biological method, or it is extracted from a biological source, or it is synthesized by a chemical procedure. So irrespective of their origin, its duty is to play an important role in the biological system. And it is very essential because a drug has to obey two important things. One is Lipinski rule of 5. In short, it will be called as LRO5, Lipinski rule of 5. And the second the second principle is ADME, right? Absorption, distribution, metabolism and excretion. So these things are to be possessed by a drug molecule, right? The first one is Lipinski. Every drug molecule should obey the Lipinski rule of 5, and it should obey ADME principle, right? For example, a company is attempting to market a drug to the global level. Means first the company has to file an application, and the application is called as NDA, new drug application, right? It has to be filed by the company who is holding the rights for marketing the drug, and this application will be forwarded to an organization called FDA, Food and Drug Administration, which is in US, and this organization will consider and will process every drug molecules, and they will give permission to market the same for the use of public, right? It should be represented, or the application has to be filed by the company. The filed application is processed by the organization called FDA. Another important thing, it is not a process which begins tonight and ends by tomorrow morning. It's not like that. There is a prominent history behind every drug discovery, right? You know, it takes nearly 12 long years for the process to complete, right? What we know is very simple. We'll go to the market, or we'll go to the drugstore or pharmacy store, and according to the prescription, the pharmacist will give the medicine, right? And this is very easy one, but there is a long history behind every drug molecule. It takes nearly 12 long years, added an investment of approximately $270 million by the company to market the drug, right? It's such a huge amount. So a long history, it refers to 12 long years, and the amount invested by the company is $270 million, right? And before the drug is introduced into the market, it has to pass through 3 phases called phase 1, phase 2 and phase 3. If a drug pass all these phases, then only it is recommended for marketing purpose. Otherwise, if it fail in the sense, the drug will not be allowed to enter into the market. Fine. We'll move on to the concept. In today's science, discovering of drug for many kind of disease plays an important role in curing patients' issue. Right? Every day we are under the threat of several diseases. Right? Nowadays, so-called corona. Right? It is threatening the people in a global level. So it is very essential and it is very responsible role to be played by a pharma company to introduce new kinds of drugs for the benefit of society. Drug institute and pharma companies always doing research with new product under bioinformatics base. Here bioinformatics playing a main role because a perfect professional from bioinformatics domain can alone able to design drugs because he know what is drug designing, and he know how to predict the structure of ligands, and he know how to predict the structure of receptors and the parameters assisting in the correlation of receptor ligand activity. All these things are known by the bioinformatician, right? The role of chemist people is to identify the lead molecule, that's it. But every other things are to be carry forward by the bioinformatic personalities, right? But so you have to be very clear about this area. What is the role of an actual bioinformatician in drug discovery process? Fine. There is a company in India, right, called the Central Drug Research Institute, CDRI, which is in Lucknow, a pioneering institute doing research on modern drugs and its activity, right? So India is a growing country with all potentials, and what is need is a professional from bioinformatics domain. So you people, you are the responsible people to become a drug design professional. So follow me very carefully. These researchers carried out only for their commercial purpose and beat the competition. It is the ultimate aim, right? Because everyone want to be the first, right? So they follow novel rules, novel methods, novel drugs to compete with others. Basically, we know how the medicine mingle with our life, but there are some steps to be carried out under laboratory and bioinformatics field to check overwriting, copyrights, etc. Because it is purely governed by the patents, copyrights, right? We cannot copy the formulations from other company. If one do these kind of things, it is a clear violation, so that a company is subjected to face legal consequences. So be clear that you are free from all litigations before filing a thing or before filing a patent. Your finding should be very novel. I mean, the formulation about the drug is very novel, and no part is reflected in others' product. It should be noted very carefully, otherwise you are subjected to face legal consequences, right? And the last one, if an industry forget to follow these methods, will fall under failure and the product announced by the government, right? If we do not stick on to the rules, I mean, examining the copyrights, everything, we will be announced by the government as a failure one. So before marketing a drug or before identifying a drug, it is very important for the personality or for the professional to go through all these things. I mean, patent issue, copyright issue and all, right? This is the pipeline of drug discovery. This is the pipeline of drug discovery, and it is very important one. It should be the same diagram should be depicted by the students in examination papers, right? So try to memorize the flow chart very carefully. And what is actually the biological data? It is all about the information regarding the molecules, right? The compound to be suggested as a drug. And target here refers to the receptor molecules, target otherwise refers to as receptors. So what are receptors? Receptors are proteins expressed on the cell surface, right? What is the role of the receptor? A typical receptor is the only channel through which a cell communicates with other cell, right? So receptors are communicating channels between cells. So first it is very necessary to identify the receptor for the specific drug we are applying. Second thing is mass screening. What is meant by mass screening? Because from a source of drug we could isolate millions of compounds. It's very common. Millions of compounds, it's very common. But among the millions of compounds, which one is the right compound to bind with the receptor? So it is a time consuming process. It takes more time to finalize the product, because mass screening is done mainly for the purpose of lead compound isolation. Another important word, target refers to receptor, right? We'll take it as R, and lead compound refers to the drug molecule. Two technical terms are used here. Target is the receptor, lead is the drug. So to mass screen, why do we need to do mass screen? Because to isolate specific lead compound. And after the mass screening procedure, we are going to validate the target, and we are going to refine the molecule. For after the refinement, the chemicals or the lead molecules are biochemically characterized. Biochemically characterized. I mean their structural compartments, their atomic numbers, what kind of atoms are constituting the molecule. All these things are characterized through biochemical process. Finally, after the design process, the drug is synthesized in a chemical way, or it is as such extracted from the biological source, maybe a bacteria and fungi, right? After doing all these things, what is the next step is clinical trial. See, we have identified the target, the receptor. We have identified and purified the lead compound. Everything is over. Then go for evaluation, whether the drug designed by the professional is good or not, right? This is a very important procedure, and there are three phases before the drug is being marketed into the public. Phase 1, phase 2 and phase 3. Initially, the toxicity are tested in animals, because no country will allow the usage of drugs directly on human beings, right? First, we should apply and evaluate the toxicological profile of the drug in cell models or in laboratory animals. Once the result is positive, then the people will take the drug into the market, right? Next one. There are 5 steps involved in drug designing. There are 5 steps involved in drug designing. One is target identification, target validation, lead identification, lead optimization, and pharmacology, preclinical pharmacology, right? As I told you before, this is a very important question. Try to follow the points carefully, right? Let's see the steps one by one. What is meant by target identification, right? This process is a chemistry based process. It is 100% chemistry based process, which means produce compounds for screening, right? In drug discovery process, imagine an inverted triangle, right? Inverted triangle, it starts with the broad upper area and smaller lower area. In this way, from 1 ton of biomass, we could be able to isolate few micrograms of lead compounds. See the difference? The biomass is 1 ton and the resulting product with activity is 1 microgram. Which is less than 1 milligram, right? So screening is an important step. right? The next one, approach of identification includes protein expression, structural and functional studies, and study of biochemical pathways. After identifying, after screening process, these are applied for its activity. They are studied in a basic way in cell lines. How it influences with protein expression, how the newly synthesized drug molecules will influence with structural and functional studies, everything. There are three other techniques or three more methods which are available for the identification process. One is sequence analysis, positional cloning, and cDNA library generation. Right? I told you before, targets are receptors, and the receptors are expressed on the cell surface. After designing a drug, our target is the receptor, or our target is the proteins expressed on the cell surface to arrest the progression of disease. Right? Because I told you, receptors are only channels which communicate the cells to the external environment. Right? So be clear, a receptor is very key molecule to be accessed by a drug for modulating the functions of the cells. Right? Next one, what is called a target validation? First is identification procedure, second one is validation. Validation, after identification of targets, it should be validated. It should be validated whether the identified molecule is the right one involved in this particular disease. Right? Because there are thousands and thousands of receptors are there on the surface of a cell. Right? Don't expect only one receptor or two receptors are there, because there are thousands of receptors. So from the 1,000, as a professional, you have to identify the receptor and its corresponding disease. Right? It is the process of validation. Target validation is a process to confirm either our drug will affect the specific target to get a biological response. Because cell is a biological entity, it will respond. Right? It will respond clearly like human beings. Right? It will respond very clearly. So based upon the drug, a cell can respond to the external environment. Fine. Chemogenomics, similar to this process, to check all possible drugs to all possible targets. The procedure by which this task is executed is chemogenomics. Target gene disruption, TGD. You can expect this expansion in 2 mark. What is TGD? Target gene disruption refers to several methods of target validation. Right? It relates to production of knockout or transgenic animals to study the effect of removing a particular putative molecular target. Another important term referred here is knockout. Right? What is knockout procedure? A very common thing in every book, but you should understand what is the knockout procedure. To identify its function, the actual gene is either disrupted or its function is switched off. Right? See, in a newly constructed home, there are 20 switches in the hall. You want to know which switch correspond to a function. Which one is for tube light, which one is for fan, which one is for washing machine. Right? So what we people will do, we will check on its operation. Likewise, the function of a gene can be identified by either disrupting it or taking it away from the source. Right? And this process is usually time consuming one. What is lead identification? This is actually the drug. The first one is, the prior one is called target. So far we are discussing about the targets or proteins expressed on the cells. But this one is called the lead. Lead identification, termed as a lead that must exceed a specific potency threshold against the target. Specific potency. Mark the word. Right? What is meant by specific potency? Right? The drug what you are synthesizing will bind with the specific target, not with other targets, because it may end up with side effects. Right? So it is very essential to check the specific potency. The compound used as lead can from many sources such as peptide libraries or from CHO libraries, right? We could check its activity. We could cross check its activity, right? Each lead must be screened by appropriate assay method against the target. Which drug will bind specifically with a specific target? An important thing, and this principle is very important, and this stage is very important to market a successful drug into the society, right? And the fourth step is called lead identification. Lead optimization. So what is optimization? In order to maintain the traits of the drugs, the process carried out. Traits. Trait means what? Indigenous characters. The purpose, for what purpose the drug is being introduced into the animal model, or it is being introduced into the cell line cultures, that should be obeyed by the lead compounds. So it is optimized during this procedure. And drug designing under CADD, computer aided drug designing. This is also an important abbreviation with respect to your paper, right? CADD is computer aided drug designing. In order to know the CADD and lead optimization, you should know de novo drug designing process, right? See, whatever we discuss here is an in silico procedure or a dry lab procedure. It is not a wet lab procedure, it is a dry lab procedure. I mean, you are doing all these calculations in a system, but this result is not enough, or this same result will not be expected by a researcher once it is applied in a real time biological environment, because it may differ, the response may differ. So to avoid the negative response, it is very necessary to narrow down the traits of the lead molecules, right? Preclinical pharmacology, another important and the final step in drug designing pipeline. Sampling. What is meant by sampling? Here, first study product testing with the laboratory animals. Now you have developed a product, so-called a drug molecule. Its activity is tested with laboratory animals, because as I told you, no company has the rights to test its product directly on human beings, right? There are several ethical issues are there. So it should be initially tested with animals. Then if the animal responded in a right way, the drug is carried over to the usage of human beings. Second one, product testing with animal cell lines, because there are rules and regulations. CPCSEA, an organization which controls the ethics on the laboratory animals, because we are not allowed to sacrifice many animals for a research at a time, right? It is strictly monitored by this organization CPCSEA, right? Though we are a multi-millionaire company, though we have invested billions and billions of dollars in drug discovery process, we don't have rights to sacrifice more number of animals for a drug. It is limited by this organization. And each company, each clinical pharmacology laboratory has an identity number, right? This number has to be revealed. If the company is found to be violated the guidelines given by CPCSEA, definitely the license will be terminated by the government of India. Third one, carried out in environment with government support. I mean, this is how the drug reaches the market. Fine. So there are three: sampling, product testing with laboratory animals. If the animals work in a nice way, it is allowed to test in the cell lines in the normal laboratory. If the test gives positive results, the drug is carried to the environment for human testing. Right, guys? Let's sum up the things. The drug discovery and $1 million business. Sorry, it's a $1 billion business which support the Indian economy in a great way. So you have to be very thorough about the pathways, the steps involved in discovering a novel drugs for a medicine for a disease. So you should be very aware of the steps involved in the drug discovery, and also you should understand the risk associated with the drug discovery process, right? Thanks for watching us. Thanks for being in online. Thanks for the opportunity given. Thank you.